So the first thing I'd like to do is to get you to do an exercise. So you need to be sitting in a chair. You need to be sitting in a chair. And it's helpful if you're sitting cross-legged. So sit in such a way that your right leg is over your left leg. So you're sitting cross-legged. Okay, so do that. I don't know if I can't really demonstrate because I don't have a high enough chair, but so anyway, what I'd like you to do with your right foot is to circle it in a clockwise motion. So while you're sitting, circle your leg in a clockwise motion. Now, while you're doing that, what I would like you to do with your right hand is to draw the number 6 and try and keep your foot going in a clockwise motion. Okay, so draw the number 6. Okay, hang on, start again. Okay, so your foot is in a clockwise motion. Try and keep that going and draw the number 6 in the air. Oh, did it work with anybody? Was anybody able to keep their foot going in a clockwise direction? Probably not, and I have no idea why that is, but I do know it has to do with neural integration. It has to do with neural integration and crossing over of neurons in particular places between the brain and different parts of your body. And it also has to do with learning, how we've learned, and it has to do with connections. So let's start with chemical synapses. Chemical synapses. I'll just move that pointer. It's funny, isn't it? Oh, I've got it. Do you play piano with both left and right hands at the same time? Yes, it is such a difficult thing to do that, isn't it? Yeah, it really is. When you're learning piano to do two different things with your hands, that's a very good point. Yeah, yeah. So chemical synapses. A chemical synapse is communication between two neurons. Between two neurons. The first neuron in the pathway is called the presynaptic neuron. Presynaptic, before the synapse. The other neuron is known as the postsynaptic neuron. And as all neurons do, there are plenty of mitochondria to provide energy for this process, which requires energy. So this is some terminology. There are inside the presynaptic neuron plenty of vesicles that contain neurotransmitters. We'll learn about different neurotransmitters. The space in between the two neurons is known as the synaptic cleft. On the postsynaptic neuron, there are neurotransmitter receptors. And there may also be, and are, ion channels. And on the presynaptic neuron, there are calcium channels. Calcium channels. Very neat and tidy. Well, what I'd like to do now is something not completely new, but let's try some drawing, and some writing. I encourage you to write because it's getting more and more evidence that writing is useful to learning, in that it encourages more synapses and better memory. So the synapse between two neurons. I'll try to write as neatly as possible. The first neuron is presynaptic. Sometimes also known as the sending neuron. The second is postsynaptic, also known as the receiving neuron. Now, a synapse, a chemical synapse, that is, there are electrical synapses, so I'll get to that later. A synapse may be what is known as axodendritic, axosomatic, or axoaxonic. So the sending neuron is always sending signals from the axon, the sending signal, but the receiving neurons may be receiving the signal at the dendrite or at the cell body or at the axon. So what that would look like is this. This is your sending neuron with dendrites, an axon, and the axon terminals. This is a receiving neuron with dendrites. So an axodendritic synapse will be from the axon to a dendrite. So in this case, the dendrite is receiving the signal. So that is axodendritic. Axosomatic. I know these look like funny little stick people, but trust me, they are neurons. Axon, terminal ends. So in this case, the axon connects to the soma. That is axosomatic. And the other instance is the sending neuron, the receiving neuron, the axon. That one of these signals is directly connecting, oh, it went straight, to the axon. And that is known as axoaxonic. So these are all the sending or presynaptic neurons. And these are postsynaptic. And I really hope that you're drawing this as well. You're probably drawing it better. All those art classes. You know, I did take art classes specifically to try to get better at drawing on the blackboard. But it just has to happen so fast and it's just too fast to make it really, really good. But the point gets across. So what about the number of synapses on a postsynaptic cell? So the number, the number of synapses on postsynaptic cell are variable. It depends where the cell is. It depends where it is. So in the spinal cord, so a spinal motor neuron may receive something like 8,000 or so synapses. But in the cerebellum, a cerebellar neuron may receive something like 100,000 synapses. The cerebellum is instrumental, if you remember, in balance. Yeah, so I have a question. Which is the presynaptic neuron? The presynaptic neuron is the neuron that sends the signal. It sends the signal. The postsynaptic neuron receives the signal. Strictly, electrical synapses do exist. They are more rare, but they're found where, even though synapses are very fast, but sometimes the signal needs to be even faster. And as you can imagine, that's in the heart. So in the heart, cardiac muscle, smooth muscle as well. And some neurons. And strictly electrical would be like this. Here is your presynaptic and your postsynaptic. Neurons, or postsynaptic, it might be a muscle cell. And here is a gap. Oh, no, I erased the whole thing by mistake. Hang on. Let's draw that again. I'm not going to be able to do that. I'm just going to draw it with red. This is, okay. Hang on. That is a gap junction. So the electrical signal, the action potentials just simply pass from the pre to the post synaptic neuron. Yeah. So that's an electrical synapse. Excellent. There are different kinds of neurotransmitters. Not all neurotransmitters are the same, and neurotransmitters can also be hormones. Neurotransmitters can also be hormones. There's about 100 different ones, quite a few different types of neurotransmitters, but they fall into 4 major categories. One is acetylcholine. Acetylcholine, which is formed from acetic acid and choline. So this is what it looks like here. And it is, for example, used in muscle contraction, and it's also used in the autonomic nervous system. It may be excitatory or inhibitory. So it could have both effects depending on the receptors. So that's one kind. Amino acid neurotransmitters, there's plenty of those. Amino acid neurotransmitters, glycine, aspartic acid, GABA, GABA. We'll talk more about that because GABA is an inhibitory neurotransmitter. It's one it's very important. And turns out that alcohol, which can have an inhibitory effect in the nervous system, can somehow replace the GABA. And that is why if an individual is an alcoholic and cuts off alcohol, that they may have tremors. They may have tremors because their GABA is no longer being produced. It had been replaced by alcohol. And indeed, that is how a lot of addictions work. If you replace, for example, endorphins, or if an individual does some kind of opiate in the form of heroin, for example, then that will take the place of the natural hormone or neurotransmitter that's produced. In our case, it's endorphins. So once an individual stops taking the synthetic, kind of like a hormone, of course, then it takes a while for the body to start naturally producing the same type of chemical in the body. So, okay, what have we here? Monoamines. Monoamines, they're synthesized by replacing the carboxyl group with a different functional group. And some that you've probably heard of, catecholamines, epinephrine, norepinephrine, and dopamine. Those are some catecholamines. The other kind, the fourth kind, is neuropeptides. Neuropeptides. We'll look at those in a moment. So that's four main categories: acetylcholine, amino acids, monoamines, and neuropeptides. Yeah. And so a point I wanted to make, I'll get to that in just a moment. Neuropeptides. So neuropeptides are just simply chains of amino acids. So chains of (20 to 40) amino acids, neuropeptides. So insulin, for example, is a kind of neuropeptide. They act at low concentrations. Neuropeptides, they have long-lasting effects than some other neurotransmitters. Interestingly, some are released from non-neural tissue. So for example, the gut releases some neuropeptides that cause food cravings, and some function as hormones. Now, that's a general comment. Some neurotransmitters also function as hormones. The difference is that hormones will be released into the bloodstream. So for more information about hormones and their actions, there is a good table in the book that I encourage you to look at. So for example, acetylcholine is located at neuromuscular junctions, but also the autonomic nervous system, the retina, skeletal muscles. Interestingly, it has an inhibitory effect on cardiac muscle and excitatory or inhibitory effects on smooth muscle. So you're probably wondering, what should I remember of all this? You should remember that there are 4 categories. I think it would be difficult to remember which hormones belong in which category at this point. But it's important to remember that some are inhibitory, some are excitatory, and also that some are neurotransmitters, some are hormones, but also that the action of a neurotransmitter depends not only on the kind of a neurotransmitter, but also on the kind of receptor. So remember, the 4 categories. That neurotransmitters may be excitatory or inhibitory, that they may also be, this is remember about neurotransmitters. Let's write that in the heading. May be hormones as well. I think so, and we'll talk specifically about a few of them with respect to the different kinds of synapses, and those ones you should know in more detail, but we're going to go over them in more detail. So really, 3 kinds of synapses with different modes of actions. So there's 3 kinds of synapses that you should know. An excitatory cholinergic synapse, an inhibitory GABAergic synapse, and an excitatory adrenergic synapse. So those are the 3 that we'll go over. Synaptic delay across a synaptic cleft is about 0.5 milliseconds, and the cleft itself is a 20 to 40 nanometer gaps. It's a 20 to 40 nanometer gap. And a nanometer is a billionth of a meter. So it's a very, very small gap. Let's look at an excitatory cholinergic synapse. What happens? Here's number one. This is a calcium channel. In this case, an action potential has reached the end of the postsynaptic neuron. And its function or its action is to open calcium channels. So a nerve signal opens voltage gated. Remember, action potentials cause depolarization, which opens voltage gated channels in some cases. But these voltage gated channels are calcium. C++. That's number one. Calcium has the effect of yes, calcium has the effect of triggering the release of the neurotransmitter. And in this case, this is a cholinergic synapse. The neurotransmitter is acetylcholine. Acetylcholine, which I'm just going to write as ACH, acetylcholine. Actually, I think the C is supposed to be capital. Acetylcholine, which crosses the synapse. Number 3, which is not quite where it should be. This does get recycled, so it's the vesicles are recycled. But the important thing is number 3 should actually be down here. That's number 3. So cross out that number 3 there. Number 3. So what's happening there is that acetylcholine receptors, these are acetylcholine, excuse me, sorry. Acetylcholine receptors open sodium channels. And they produce a kind of potential. Is it? Sorry, yes, Ca. Thank you, Jenny. Calcium is Ca++. Opens the sodium channels. Do you think this results in an action potential in the receiving cell or a local potential? This one action. Creating a local potential. Yes, I'll talk about insulin in a moment. So, yes, insulin is a hormone. You're right. Insulin is a hormone, but it is, as many neurotransmitters are, it is a peptide. Yeah, you're right. Insulin is definitely a hormone. So, going on to the 4th action. What's going on here? When local potentials reach, and what is the voltage that local potentials have to reach in order that there is an action potential? How many millivolts is the threshold? Minus 55. Good. So when they accumulate and reach minus 55 millivolts, then an action potential is produced. Good. So I have a question about why is it a local potential and not an action potential? Just one of the receptors opening, or even a few, are not enough. So we did talk about the summation, or we're going to talk about it if we haven't yet. We're talking about the summation of potentials and the summation of synapses, rather, and whether or not it will produce an action potential or not. It turns out it takes quite a few synapses to cause an action potential to occur. Yeah, and often there are inhibitory synapses happening at the same time. So whichever one outweighs the other one is what that postsynaptic neuron experiences. And we are going to talk about that for sure. So that, my friends, is an excitatory cholinergic synapse. Yes, so that's good. Inhibitory GABAergic. Well, GABA stands for gamma aminobutyric acid. Gamma aminobutyric acid. That's what GABA stands for. And it's quite simple the way it works. Actually, this one is quite simple. What its action is, its action is to open chloride channels. And what happens when chloride channels are opened? Chloride enters the cell. And chloride just happens to be negatively charged. It's an anion. So it hyperpolarizes the cell. So now it is less likely that the threshold will be reached. It is less likely for that to occur. So that is our second kind of synapse. The third kind is called an excitatory adrenergic synapse. And this is through a second messenger system. And the second messenger system, well, there can be multiple effects of this system. Multiple effects. CAMP, cyclic adenosine monophosphate, is triggered and may cause enzyme activation, genetic transcription, some metabolic changes due to enzyme synthesis. One important effect is also a postsynaptic potential. So the cyclic AMP may cause the ligand-gated channels to open. The ligand in this case is cyclic AMP. So the order of events is such. Here's the receptor. Oh, and you know one thing, did I already say this? That, yeah, the effect of neurons is not just because of the neurotransmitter it releases, but also the nature of the receptors. So for example, there are beta receptors, and I think I talked about this in the very beginning, beta receptors, and that's where beta blockers come in. So the nervous system, neuron transmission can be manipulated with synthetic drugs in that way. So receptors can be blocked. Neurotransmitter uptake can be blocked. So there are different methods by which to manipulate the nervous system. And it does also sometimes target receptors. So here is your neurotransmitter receptor for, in this case, it's adrenergic. The neurotransmitter is norepinephrine. So it's a bit confusing, because norepinephrine used to be called noradrenaline, and epinephrine used to be called adrenaline. So to me, that makes more sense because they are also released by the adrenal glands. But the adrenal glands, they sit on top of the kidneys, so now it's called epinephrine instead, above the kidneys. Yeah, so that's a bit of an aside. The naming of neurotransmitters. I'll bet most of these were named after the people that discovered them in the first place, but now we don't use eponyms as much. Yeah, so these are the steps. So the question was, what are the steps? As it's a second messenger system, this is the first step. In this case, the same process releases neurotransmitters from the presynaptic neuron. So the difference here is in what happens with the postsynaptic neuron. For one thing, the receptor is different. So it's a receptor for norepinephrine. So here's our little no, I'm going to make it blue, our little norepinephrine here. And that would be step 1. That norepinephrine binds. Wait, so I didn't put it in the right place. 1. The receptor is already there. So norepinephrine binds. 2, it releases a G protein. The protein itself binds to adenylate cyclase. When it binds to adenylate cyclase, phosphorylation occurs and ATP is stripped of 2 inorganic phosphate groups and becomes cyclic AMP. So that's, let's call that 4. And 5 is the opening of ligand-regulated gates by cyclic AMP. So let's write that down. 1, a norepinephrine binds to the receptor. 2, the G protein is released. 3, adenylate cyclase is activated. And when it is activated, it converts ATP to cyclic AMP and 2 phosphates. So adenylate cyclase is not an ion channel. Rather, it triggers the opening of a ligand channel. So the ligand gates open. So you might be wondering, how is the signal stopped? Of course, you don't want your signals to carry on forever and ever. That would be exhausting. So there are mechanisms to turn off stimulation and neuromodulators that modify these transmissions. So mechanisms to turn off stimulation are such things as simply diffusion of the neurotransmitter away from the synaptic cleft. So if you have a neuron there, a neuron there, here are the neurotransmitters, and they diffuse away, and that stops transmission. The synaptic knob may just reabsorb the neurotransmitter. That's another way to stop transmission. And that occurs in the case of amino acids, especially, and monoamines. What happens to them? They get brought into the cell by endocytosis and then broken down. So by endocytosis, and then they're essentially recycled. And the third kind of mechanism is, this happens to acetylcholine in particular. There is an enzyme called acetylcholinesterase. Acetyl, some people say acetyl. Acetylcholinesterase. And it degrades acetylcholine right in the synaptic cleft. So neuromodulators, this will be our last thing before we break. Neuromodulation. So that was just what we just talked about. That was all about cessation, which means stopping the signal. Neuromodulation is modifying the signal. And there are different ways of doing it. One is to increase the number of receptors or decrease the number of receptors. So you can raise or lower the number of receptors. And that occurs as a response to signals from elsewhere, or it occurs over time due to desensitization to particular neurotransmitters, for example. Also, neurotransmitter release can be altered. Alto neurotransmitter release. Synthesis or breakdown. So I thought that was an example. There's not an example there. Let's see, what's a good example? Okay, a neuromodulator example is nitric oxide. So nitric oxide diffuses into the postsynaptic cell and activates a second messenger pathway, which has the effect of, for example, of triggering inhibitory signals to smooth muscle. So relaxing smooth muscle and thereby increasing blood flow. So this is the principle on which Viagra works. So Viagra, which is a drug for erectile dysfunction. So Viagra triggers or somehow enhances the production of nitric oxide. Another example would be, say, endorphins. Endorphins are modulators as they block pain. In spinal neurons, so the neurons that transmit pain signals are blocked. So that's why it's so great to exercise. Endorphins have an amazing effect on the body. I don't know if you've ever heard of a runner's high. You know, somebody runs a lot and they release so many endorphins that afterwards they just feel fantastic. They're not feeling any pain because the endorphins are blocking pain transmission signals. So that is all I wanted to say for the moment. And now I'll sign off.